Welcome back. It's Intro to Physical Anthropology. I'm David Leitner. I am your instructor, and today we're going to talk about mutants. Mutations are incredibly important to evolution. They are the stuff of variation. They are where variation comes from. And without variation, natural selection has nothing to work on. Evolution can't occur without variation. So we need to understand what mutations are and what the implications are. So without further ado, come on, there we go. Mutations, as we've established before, are just changes to the genetic code during replication. That's it. That's it. They could come from radiation. They can come from heat. They can come from all sorts of sources. But the point is that it's a change to the genetic code. Now, there are different ways this can happen. There are different kinds of mutations. Each of them has a different implication for how it impacts the function of a gene. But three things to keep in mind. Overall, this isn't three. They all pertain to this. The vast majority of mutations are benign. Okay? You're walking around with hundreds, if not thousands, of mutations right now that you'll probably never know about. Okay? Unless you have your and your parents' genetic codes compared. This can be for several reasons. They're benign for several reasons. The mutation might occur in a non-coding region of the DNA. Not every part of our DNA codes for proteins. Other parts seem to do other things that we're not real clear on yet. It's still, I won't say it's a mystery, but it's certainly something we're actively investigating. If they happen in those regions, it seems like they don't really have much of an effect. The mutation could occur in a coding region, but doesn't change the amino acid sequence of the gene it affects, or the protein that the gene makes. And even if it does change the amino acid sequence, that different amino acid might not change the shape and chemical properties of the protein in the end. So it could be either of those. So yes, it's a change, but it's kind of not a difference, if you get my drift. Finally, you can have mutations affect you, but if that mutation is not under selective pressure at the moment, it's a mutation, but it doesn't really mean anything in terms of evolution. You may or may not pass it on, but it's not going to be under selective pressure, so it will naturally increase over the course of time. So it's really, when we talk about mutations, people want to talk about bad mutations and good mutations, and it's really natural to talk in those terms. But it's probably better to think in mutations in terms of their relationship to the environment. Are they adaptive, or are they maladaptive or non-adaptive? Okay, if it's maladaptive, it affects the ability of the protein to function in such a way that fitness, that is, one's ability to pass on their genes to their grandchildren, that is more than one generation, is reduced. So reduction in fitness can occur from maladaptive genes. Adaptive genes, on the other hand, will increase fitness. But again, remember, this is in relation to a particular environment. If the environment changes, what was a maladaptive gene can now be an adaptive gene. So this sort of label is context dependent. Many, in fact, most mutations are neutral. They make no real effective change whatsoever. And it doesn't change fitness, even if it's a very visible one. Maybe I grow blue hair, but it doesn't affect, but no one in our species has the ability to see blue. Doesn't change thing in terms of my ability to reproduce. So that's the key here. Remember, with mutations, for it to matter in terms of evolution, it has to be passed on generation after generation, and there has to be a selective pressure. So, the most common kind of mutation that we can talk about is a point mutation. Okay? Sometimes these are called single nucleotide polymorphisms, or SNPs for short. They are exactly what they say. They are a single point. One point on the genome that changes. A single letter changes. So you could have CATAGC, as I've listed here, change to CATGGC. There you go. A great example of a point mutation at work is the gene for the hemoglobin protein. The disease sickle cell anemia results from having a version of the hemoglobin protein gene that creates red blood cells that, although they can carry oxygen once they release their oxygen, instead of maintaining this nice donut shape on the left-hand side, they collapse into this crescent shape on the right. Those crescent shapes are really good at clogging up the tiny blood vessels and capillaries and so forth in the body, especially in the lungs where oxygen transfer occurs. They get stuck in there and they clog things up. Luckily, if you have one functioning copy of this gene, you're going to be okay. Now, the difference in the alleles is 1 out of the total 438 bases that make up the gene. This is a very small gene. It's only 438 base pairs long. And only one letter, a change of a T to an A, is responsible for this. It basically inactivates the protein that it's supposed to create. Now, sickle cell anemia is a recessive trait. So you have to have 2 copies of it, like I said, to actually get sickle cell anemia. But if you are heterozygous, that is, you carry one normal copy of the HBA gene, and one sickle cell copy, that would be the HBS gene, then you see no signs or symptoms of the disease. You go on your merry way, not even knowing necessarily that you carry this gene. That's important because a person who is heterozygous still has a normal gene that can take up the slack for the defective one. But a person who is homozygous for sickle cell, that is, they have 2 copies of HBS, they will develop sickle cell anemia because they have no normal copies of that gene to compensate. Now, the hemoglobin gene is inherited in a Mendelian fashion. That is, it's a strict sort of, you either have it or you don't situation. And the sickle cell gene, HBS, is recessive. So if 2 heterozygous parents have children, what do you think the chances are that their children will have sickle cell anemia? Go back to thinking about Mendel, thinking about Punnett squares here, and take a moment. Hit pause if you need to. Okay, so if 2 heterozygous parents have children, what are the chances one of their children will have sickle cell anemia? Well, let's do the math. The genotypes are distributed as follows. You've got 1 homozygous dominant who will have 2 normal copies. You have 2 heterozygous individuals who will have 1 HBA and 1 HBS. And finally, you will have one homozygous recessive individual who will be HBS. That individual will end up developing sickle cell anemia. The outcome is 1 in 4. 1 in 4 of their children is likely to have sickle cell anemia. Sickle cell anemia is deadly without treatment, and treatment is relatively recent thing, sort of in the last half century or more. And without treatment, most individuals die as children. With treatment, people can live long, healthy lives. Although their overall lifespan is still shortened, they can reach adulthood. So, now, this is a weird gene. This is the sort of thing where you would think, if people die so quickly from it, why doesn't it disappear? I mean, it's clearly maladaptive, right? If you have the HBS gene, even if you're homozygous recessive, then you won't get to have children at all without treatment. And if you're heterozygous, well, 3 of your children are likely to survive, but 1 of them will die. So compared to the homozygous dominant individual, who will have 4 out of 4 living children, the heterozygous individual has less fitness. They are less likely to pass their genes on to grandchildren. And given a normal context, that's exactly what we see, in fact. So the homozygous dominant individuals have an advantage over the heterozygous individuals. And HBA tends to dominate. Interestingly, though, there's a film on the website that I think you should watch. It's about the discovery of this fact and the science behind it. When you map out places in the world where the HBS gene is more abundant and sickle cell is more abundant, you very quickly see a pattern. They are almost exclusively around the tropics and almost always near bodies of water. In other words, they are close to breeding grounds for mosquitoes. Mosquitoes carry a disease called malaria, and as it turns out, the parasite that causes the disease, which is passed to you when the mosquito bites you, cannot infect sickle cells. And so it can't get as good a foothold on the bodies of heterozygous individuals. They have an advantage over homozygous dominant individuals in areas with malaria, because it is much more likely the children of a homozygous dominant individual will die of malaria than the children of the heterozygous individual will die of malaria. So heterozygotes have an advantage in those areas. And sure enough, we see a much higher prevalence of heterozygous individuals and a much higher prevalence of the HBS gene overall. It is a fascinating story. I really encourage you to watch that film in the optional section on Canvas. Right. Different kinds of mutations. You don't just have point mutations. There are others as well. The top one here is a substitution mutation. This would be like a common thing for a point mutation, but it could also be a sequence of genes that get replaced as well, substituted. And it's simply, where it was GA before, it's now GA. They replace the first A with a G. That's it. You can also have a deletion where one or more of the letters in the middle are removed. So here they remove the middle AG. I'm just looking at the top line to make things a little easier. They remove the middle AG. And so now you've actually shortened the sequence, the genetic sequence here. You can have the opposite. You can have an insertion. So here they've inserted another GA into the middle. And finally, you can have an inversion. An inversion is when they copy the wrong side of the DNA when they're replicating. So the left side goes to the right and the right side goes to the left. I'm saying right and left, but you get my picture there, right? It's just flipped. That's it. You can also have them flipped the other way as well. So instead of being GAGA, it would be AGAG. So there are lots of different ways this happens. There's also, as we'll talk about in a minute, something called a repeat as well, where you just get multiple insertions of the same thing over and over. Okay, so with insertion mutations and frankly deletions as well, you can have what's called a frame shift error. A frame shift error is what happens when you have a mutation that affects a multiple of 3. Anything other than a multiple of 3 base pairs. Because 3 base pairs is codon, right? That's a complete codon. So if you have a mutation in 3 base pairs, you have a complete codon changed, right? The top row here is the normal sequence, and they go from the codons to the amino acid sequence, and you can see what it looks like. Well, with a frame shift error, so anything other than 3 codons, that means, if you look, you just inserted 1 T into the second codon. That shifted everything over by 1, which means now the codons read completely differently. Instead of Glen Pro Thra, it's Glen Ser His, right? Now, if instead you insert or delete a complete codon, you don't get a sequence that shifts completely. Instead, you just insert an additional amino acid. So Glen Pro Thra becomes Glen Fe Pro Thra. I think that's really interesting. So the degree to which the protein will be changed kind of depends on how much the mutation itself changes the sequence. Okay, now I want to talk about trinucleotide repeats. There's a whole class of diseases caused by these. A trinucleotide repeat is, well, trinucleotide means 3 nucleotides, and what do we know 3 nucleotides are? A codon, right? So it's a repeat of a codon over and over and over and over again in the DNA sequence. Now, the interesting thing about trinucleotide repeat diseases is oftentimes their severity, even their onset, is dependent on the number of repeats in the sequence. So to give you an example, Huntington disease. Huntington's disease is a neurodegenerative disorder. It is autosomal dominant, so you only need 1 copy of the Huntington gene or allele, and the trinucleotide that is repeated is CAG, which codes for polyglutamate. Okay, or sorry, polyglutamine. And essentially, it's expanding this sort of repeated polyglutamine. Trying to say that fast, that's not easy. These CAG repeats, basically, on chromosome number 4, tell you whether the person is likely to develop Huntington disease later in their life, because it usually comes on a bit later, usually middle age, maybe a little earlier, maybe much later. It depends on the number of repeats. Between 10 and 35 repeats, no Huntington's disease. Between 40 and 180, you have Huntington's disease. And in addition to that, there's a relationship between the age of onset as well as severity of the disease and the repeat length. Here you can see on the left-hand side we have age of onset of the disease, and along the bottom we have the number of repeats. And you can see for 40 repeats, you're over 60 before it kicks in, whereas at 80 repeats, you may not get out of your teenage years. So, so that's an interesting sort of effect of a mutation. So mutations are a lot more than just sort of like, oh, we changed a letter here, changed a letter there. There's a lot going on. You can have entire chunks taken out or inserted or turned around that change things around. Or you can just have like this stuttering repeat that creates creates a, in this case, deadly condition. Now, in all but one pair of chromosomes in the human genome, both members of the pair are homologous. That is, they have the same genes, maybe different alleles, but the same genes in the same places as the other one in the pair. But the sex cells are different. If you are a woman, you have a homologous pair. You have two X's. But if you're a man, you don't have a homologous pair, because the X and the Y carry a different set of genes. One of the genes on the Y that doesn't usually show up on the X is the SRY gene, which is the gene that triggers the formation of male genitalia in the womb. It has to switch on at the right moment in the womb for that to happen, but that is, that is one of the key differences between the X and the Y genes, X and Y chromosomes. By the way, that is just a piece of how sex is determined, and is one of the reasons why we won't get into it in this class, but in cultural anthropology, I often teach about the fact that even, we teach that sex and gender are different in that sex is biological, but even sex to some degree is determined by culture, because there is such a spectrum of indicators of sex that we have to make arbitrary decisions when we make up categories of sex. And so there's more than two sexes in humans and in other species as well. But I won't get into that here, but it's important to understand that. Okay. Examples of X-linked diseases. So when you have an allele that causes a disease that happens to be on the X chromosome, the disease pattern is really interesting. Okay. Some common examples: hemophilia, which is the absence of clotting factors. It can exceed varying levels of blood loss depending on how severe the disease is. Lesch-Nyhan syndrome, red-green color blindness. Okay, these are all examples. Now, the thing about X-linked diseases is it matters which parent has the disease and whether they're a carrier or affected by the disease. So in this case, the first two examples I'm going to give you here are an X-linked recessive father with an unaffected mother. Okay, so you can see that he has an X-linked trait. He has one copy of this gene that's on the X chromosome, but he does not have a second copy to make up for it on the Y chromosome. Okay, remember, if you have, it's it's recessive. So if he only has one gene, one copy of it, he's not going to pass it on. Or he's going to pass it on, but his children are not going to be sufferers. At most, they can be carriers for the disease, as you can see here. And only the daughters, because men don't contribute their X chromosome to boys, to sons. They have to, they're the only one who has the Y chromosome, the father is, so they have to contribute that. So it's only the daughters who will inherit that gene, but because they have a normal version of the gene as well, because they have two X chromosomes, they will only be carriers of this trait. They might pass it on to their children, but they will not be affected by the condition. Now, if you have a carrier mother, that's one of these daughters here, right? And she has children with an unaffected father. So he does not have a defective version of this gene. Their children will be a combination of two unaffected children, whether or not they're girls or boys won't matter. A carrier child, and finally a son who is affected. Now remember, these are probabilities. So there's a 50% chance they will have an unaffected child, 25% chance that they will have a carrier daughter, and a 25% chance they will have an affected son. Now, he's affected because he got his X chromosome from his mother. And his Y chromosome doesn't have another version of that same gene to make up for the defective gene that he inherited. Now, when you have an X-linked dominant trait, things look even more different. With an X-linked dominant trait, the mother passes it on, but she doesn't pass it on to her daughter as carriers. She is affected by the disease, number one, because it's dominant. You only need one copy. And her children will be affected by the disease, whether they're boys or girls, because since it's dominant, it doesn't matter what the other gene does, it's still going to be affected. And if you have, and finally, in the case of an X-linked recessive affected father, as we saw before, if you compare the two, it's a very different looking outcome, right? So X-linked disorders make things very complicated. The main thing to know is only the mother can pass on the disorder in this case, because men do not pass on their X trait to their sons. Okay. All right. Thank you very much again, and have a great evening. I hope you are well. Please take care of yourself, and I will see you again soon.